Welcome to Study Loop.
About the changes in a person's blood after receive a vaccination, and the graph basically depicts the antibody count and shows that after vaccination was starts at time point 0, that the amount of IgM and IgG, which are just two different types of immunoglobulin antibodies, are drastically increased in response to a vaccine. And so we want to explain how that occurs. So a vaccine basically contains antigens or an inactivated pathogen. So it introduces a foreign antigen that can trigger the immune response. So how does that happen? So basically we have an antigen presenting cell that detects the antigen, the foreign antigen, and it presents the antigen on a class 2 MHC molecule, which is then recognized by a helper T cell. So this helper T cell really serves as the middleman that activates both humoral and cell-mediated immunity through both B and T cells. But one specific example, if we're talking about antibodies, is that they trigger the release of cytokines that induce the proliferation of a B cell that is specific for that antigen and triggers the production of plasma cells that produce antibodies. So let's just see which statement is best true about the molecular mechanisms behind the vaccine. So A states that the vaccine introduced antigens specific to a pathogen into the blood. These antigens are moved to the cell surface of antigen-presenting cells present in the blood. Receptors on helper T cells bind to the antigens present on the antigen-presenting cell. This direct cell-to-cell contact initiates a series of events that leads to the production of antibodies by B lymphocytes. So that all sounds correct. So let's see whether the next statements are true. B states that the vaccine introduces antigens specific to a pathogen. These antigens bind to the receptors on the surface of T cells. So no, they are bound to the MHC class 2 molecules on antigen-presenting cells, which are macrophages and dendritic cells. They do not directly bind to the receptor on helper T cells. So that's B is incorrect. C states that the vaccine introduces antigens specific to a pathogen. The antigens are moved to the cell surface of antigen-presenting cells, and this direct cell-to-cell contact initiates. So no, just because these antigens are moved to the surface of an antigen-presenting cell does not directly lead to B cell proliferation. It is the helper T cell that really helps mediate that. And D states that the vaccine introduces antigens specific to a pathogen into a person's blood. These antigens are moved to the cell surface of antigen-presenting cells in the blood, and receptors on helper T cells bind to the antigens present on the antigen-presenting cell, true, and it activates the complement system. So no, the complement system is the production of proteins that enhance phagocytosis, but it does not explain the increased antibody count. It is the activation of the helper T cell that triggers the production of antibodies that really explains the increased antibody count that we see in the graph on the question. So therefore, the correct answer to this question we can say is A.